thanks for kind of introduction. And so thanks for giving me this opportunity to share my recent work. And this work is just published in this year. And this is about a platform that is aimed to help the drug discovery for the orphan GPCR. Okay, so well, this is the GPCR 101, and I will just go through that quickly. And GPCR is the largest and most diverse group of membrane receptors in eukaryotics, and it plays a role in many functions. And in a survey, that 60% of drugs in the development stage target human GPCR, and also 36% currently marketed drugs also target human GPCR. And the first links between the cancer and also the GPCR was first discovered in 1986. And after that, a strong association with the tumor growth and metastasis has been found in many studies. So despite the strong potential of the GPCR as a target of cancer therapy, but only a few anti-GPCR drugs were invented and commercialized for clinical use. So there should be a very large space to explore the remaining GPCR for the cancer therapy. But if we would like to apply the AI for the GPCR drug discovery, then we may face some problem. The first one is that when we think about the AI in drug discovery, we usually comes out with the idea that we should have a protein structure, a crystal structure. With the prettiest protein structure, then we may use the protein ligand docking software or use the deep learning algorithms to predict where the compound will bind to the active site of the protein structure. And for another approach, the artificial intelligence approach, that we may need a set of ligands that we have its activity and also its structure for that GPCR. And we collect all the data and we can apply some AI algorithms to build a prediction model, try to predict the novel structure and try to predict the effects of the chemical on the target GPCR. But when we look at the largest GPCR database, the GPCR-DB, then we find that among all those 800 or 900 human GPCRs, there are only 216 GPCRs with ligand information. That means most of the GPCRs, we don't have the data for building a reliable or even not a reliable AI model because we don't have data. But you may think about that, well, we now got the AlphaFold tech, and everything may be resolved, right? So for the orphan GPCR, there can be two ways to develop the drug using the AI. One is that we can predict the protein structure, and then we use the protein structure to design the corresponding drug. Or we will need another approach in contrast to the traditional AI model to have some prediction. So this is just show you that the orphan GPCR has very tremendous potential for the cancer treatment. So there are several papers published in recent years. And so if we want to apply the drug discovery for the orphan GPCR for their potential to treat cancer, then the first thing, as I said, you may think about the AlphaFold because the AlphaFold is currently the most accurate program to predict the protein structure. But this is a case that shows you that this is a GPRC5A that is also considered as an orphan GPCR recently. And as you can see, the predicted structure has very low confidence in the structure, especially in the loop area, and also the extracellular area. So that means if you apply this structure for the drug discovery, then you may get the wrong answer, and you may just like try and error and get a random guess. So AlphaFold is not the solution, especially for the membrane protein, especially for the open GPCR, because all the open GPCR often associated with a property that is very difficult to be crystallized, solved its protein structure. So we are thinking about the other way, that whether we can apply the AI algorithms to build a model for the open GPCR, even we don't have its legal information. So we check the property of the GPCR, and luckily we found that the GPCR has they have shared some structural features, like for example the activation engage all membrane helicase, and also the transmembrane 6 and the transmembrane 3 are universal helix switches. And so based on those observations, we have an idea that there can be some information that can be shared between those binding patterns of the GPCR and its ligand interaction. So instead of the traditional machine learning algorithms that train a model for one GPCR at a time, then we think that if we can utilize the conservation nature of the GPCR and its ligand interaction, then we may be able to develop a transfer learning algorithm that can learn the knowledge share between the GPCR ligand interaction, and then we can transfer the knowledge to the open GPCR. That means we can build a unified model for all GPCR rather than just one by one. So this is the idea that we have proposed, and that is to collect all the available information from the GPCR DB. It contains 424 proteins and with this large number of bioactivities. So if we can have a model, an algorithm to learn from this large data set, then we may be able to apply the knowledge learned from those GPCR and apply to the open GPCR. Okay, so this is the problem formulation in our study that we decompose the problem into two parts. One is the protein sequence, and we apply a similar strategy as AlphaFold that we use the multiple sequence alignment to try to get the conservation information from the GPCR protein sequence. And then the other part is the ligand part. Then we extract the protein fragment information and then encode into a 1024 bits feature vector. And also we calculate 1,444 descriptors, including the lipophilicity and also the molecular weight. And we designed one additional feature is to indicate whether this is an agonist or antagonist. So we are thinking to build two types of model. The first one is that we combine the protein feature and with the ligand feature, and we build two models, each for one for agonist and one for antagonist. And then the other is that whether we can combine all those information, including agonist and antagonist, into one. So this is the system flow for our study, and that we extract only the human GPCR data, and we separate the data set into a training and validation set, and we filter out 16 proteins with only less than 8 ligand information to be considered as the orphan test data set. Okay, then. So this data set was used for the training and for the validation, and this is for the testing and to simulate the situation when this model applied to the orphan data set, the orphan receptor. And the algorithm behind this is an ensemble model based on 5 models, including the neural network and the state-of-the-art algorithms. Okay, so this plot shows that the multitask learning algorithm did improve the performance to lower down the mean squared error compared to the single task one. So, well, there might be some shared knowledge learned from each other. And also, in the case of antagonist, the multitask learning did improve the performance compared to the single task one. And if we further combine the agonist and antagonist, we get also the best one with the lowest mean squared error and also the highest correlation coefficient. So, well, in these results, we may know that the shared knowledge can be learned, and also the simultaneous learning of both agonist and antagonist data can further improve performance. After that, then we would like to make sure that the model actually learns something from each other. So we did 2 experiments. One is the feature truncation, that we remove part of the sequence and to see what happened. And we find that there is one part, the third part of the sequence contribute most to the multitask learning. So, well, this model did learn something from the other GPCR. And also we did the feature permutation test. That is to randomly substitute the residues with the others. And we found that the part 1, the first part, contribute most. And that makes sense because the N-terminals always has already been approved to be the most important part for ligand recognition. Okay, so after that, then we would like to identify the key features because this is the only way to check how the model works to eliminate the unnecessary features to know how actually they recognize the ligand. So we apply the feature selection algorithm and we identify 162 protein features and also 38 physical chemical properties, and it remain the same performance. And when we decompose all the residues, then we found most of the key residues located in the N-terminus. And then, well, so the multitask learning did learn something from the other GPCR, and also the key residues are located in the N-terminus. So next step, we would like to check whether the knowledge sharing mechanism can be applied to any of the proteins, or there should be some limitations on the application. So we take the similarity of the testing protein sequence compared to the training data set as an indicator, and we found that when considering the full length of the protein sequence feature of the 162 feature, and we found that the decrease of the mean square error correlates with the similarity of the protein sequence. That makes sense because the similar protein sequence of the GPCR may not share the same feature of the other GPCR in the training data set. So that gives us a very good indicator to show the limits of the prediction model. Also, that can be a confidence score of the prediction. So then we applied the prediction model to Orphan data set, and this is the results. That if we apply the original model, we get a higher mean square error, and if we apply the similarity threshold, then we get a nearly perfect prediction results. And in the review of our article, then we find a paper that published in the Nature Chemical Biology. They identify two ligands for the open GPCR. So we use that as an independent test, and we found that the prediction value of our model is really close to the experimental one. Okay, then we try to understand whether the identified key feature, the key residue, is really meaningful in the protein structure. So we map the key residues into the protein structure, and we find that according to those four protein structure, the crystal structure, the identified key residues are all close to the ligands. So well, that give us more confidence on this model. Okay, this is another experiment outside of the paper. That is a real case that implemented in our institute. We collaborate with Dr. Hong and Dr. Lee, and we aim for the drug discovery of GPRC5B. And then in this case, for the agonist and antagonist identification, we got like 1% and 6.6% hit rate for the agonist and antagonist identification. Okay, so this is a summary that in this study, we show that there are some shared knowledge among the GPCR, and that we can use this information to build a unified model to make the prediction of ligands for the open GPCR possible. And we also identify the key features, including the residues or the physicochemical properties, and we make some validation on the open set, and we did some two independent tests to show the efficacy of the platform. So we consider this is a very useful platform for open GPCR drug discovery. And actually, this is not the end of the project. We are continue to improve this algorithm, and this figure shows that we are trying to incorporate the generative algorithms to further improve the prediction performance, and we see some light on the further improvement. Okay, so if you are interested, you can find a paper published in Chem Informatics, and well, that's all. Thank you.
Thank you. Any questions from audience? No one has any question? All right, I guess everybody would like to have a break. Thank you. Let's thank the speaker again. Thank you.
Thank you.